Welcome to 2022. And I am going to talk to you about a postdoc project I've been working on these past few years on internal states and how they influence behaviors in C. elegans. And so I am a postdoc at the Salk Institute, which is shown here in a dreary day, and I use the pronouns she, her. Before I begin, I would like to acknowledge that I live and work on stolen land. So this research was performed at the Salk Institute, which is on the unceded and traditional territory and homelands of the Kumai people. These people continue to maintain their political sovereignty and cultural traditions. So I'd like to pay respect to the Kumai people and their land from the past, present, and future. If you're interested in the native lands that you might have grown up on or currently work on, check out native-land.ca for this really great map. I recently did that for my hometown and found out that my hometown of Port St. Joe, Florida, resides on the lands of the Muskogee and Creek people. So I grew up in the middle of nowhere in a very rural, isolated part of the world in a swamp. Very resource limited family and town of about 3,000 people and a total of 2 stoplights. As a child, I spent a lot of times outdoors because there was a lot of outdoor engagement, and I became really interested in how all the living things that I was surrounded by worked together to make the ecosystems that I romped around in as a child. And sometimes I absolutely loved these moments, scooping up bugs with my dip net and carrying them around in this strange little necklace. But sometimes I really wasn't having a good time. Maybe I got bit by a bug, maybe my parents were mad at me for spending too long in one puddle over another. And so if you're early on in your research, and maybe you're watching this recording, think about these moments, keep track of them. And as we're growing as people, I think it's really important to keep track of the moments that we feel like joyful, happy Molly with her dip net, and try to avoid the times when we feel like this. And so one thing I truly enjoy is sharing my research and my passion for mentorship and engagement with students to others. So I'm really delighted to be here today to share with you my joy. So I started at my career at North Carolina State University. I was very fortunate to receive a full paid scholarship to attend. I have no idea what I would have done without this opportunity. And so I'm very grateful to Jacob and Betty Beelan, who started a scholarship for people from my rural area. And I studied chemistry because I was interested in understanding the mechanism through which biology happened. And I had been told that chemistry was the language of biology. So I thought, great, if I understand chemistry, I'll understand biology. That is obviously not totally the case, but it did give me some really great groundwork. And with the help of 2 amazing mentors, I started doing research in biochemistry and genetics, which really let me get into that language of biology that I was so much seeking. And so with these 2 mentors, I applied to Duke University to get a PhD in genetics and genomics. And a PhD was something I had never even heard of prior to going to college. I hadn't even heard of a PhD until I was maybe my sophomore year of college. So with these mentors and a few other amazing teachers, I learned about this trajectory and successfully applied and got a PhD. So while at Duke University, I became interested in host microbe interactions, specifically the pathogen tuberculosis and how it affects its host of a zebrafish model. And through my interest in host microbe interactions, I started my postdoc at the Salk Institute a few years ago and really wanted to keep using transparent model systems to understand the underlying mechanisms of really cool aspects of biology. So I transitioned from immunity to more neuroscience, but I'm kind of a jack of all trades and admittedly master of none. And now I hope to use all of my past experiences and passions and joys to pursue a career as a professor at a primarily undergraduate or undergraduate focused institution. So if any of these moments seem interesting to you or these paths, please feel free to email me. And if I think I see most of the participants are people with PhDs or actively pursuing them, but if you're watching this and if you don't know, PhDs and many master's degrees in STEM are free and you get a stipend. So if financial burden is preventing you from achieving your goals, know that you can actually get paid to play around and do research. And again, if you're unfamiliar, a postdoc is a training position between your PhD and another job. It's not an end goal, it is a transitory position, but it really lets you get more training, maybe switch fields, maybe get a few new skills. Just so you know, Jacob and Betty Beelan were the people I mentioned who provided me with my funding to go to college. So I'm going to begin very broadly with my research topic here, that animal behaviors are context dependent. And I'm going to talk to you about three main contexts, flavors of context that I like to think about. One is just organismal or evolutionary context, and this is the presence or absence of a structure. If you wanted to study flight, you would have to look at an animal that had wings. So for today's talk, we're going to talk about a C. elegans nematode worm, which I'll introduce on the next slide, and its intestines and its neurons. So great, it has those structures. Next are environmental or external states. And so these are things we probably thought about as we left our house. Like, what's the temperature? Is it going to rain? And it changed our behavior. Were we going to walk to work? Are we bringing an umbrella? And luckily for us, in a controlled laboratory environment, we're keeping most of our environmental or external states constant. And today I'm going to spend most of my time talking to you about internal states, which I think are super interesting and sometimes overlooked when we think about animal behavior as we're experiencing it as animals ourselves, which are our age, our circadian rhythms, our immune status. And what I'll talk about today is hunger and the composition of the gut microbiota. So everything I'm going to show you today is with this little guy, Caenorhabditis elegans, or C. elegans, which is a nematode worm. And they've been studied for the last 80 years because they're such good models for a lot of basic biological processes. They actually eat bacteria, so they slurp it up, grind it, and it goes through their digestive tract, which is just a tube-like intestine. They poop, everybody does it, they do it too. They reproduce. These animals exist primarily as hermaphrodites, so they have both sperm and egg, and they grow old. Their tissues start to decay, they function less well as they age, and they have a pretty short lifespan. So a lot of research is actually done on how these animals grow old and the mechanisms underlying aging. But I'm going to talk to you mostly about how they can make decisions. So they have 300 or so neurons. A lot of them are in their head. We can call it a brain. It's not quite a brain. And they're capable of deciding where to go and traversing their environment. And so I'm going to talk to you about their decisions. And to remind you, all of this is within 1 millimeter of an animal. So everything that I'm showing you today has been done under a microscope. So these 300 plus neurons that make decisions are capable of somewhat complex behaviors. And what I'm going to tell you about is called sensory integration. And I'll give you an example of us performing sensory integration as a population of people who maybe want to go get some ice cream. So some of us would decide, let's go together and let's go get some delicious ice cream. But now, if say there was something between us and the ice cream, like a pit of snakes or someone yelling at us, maybe we wouldn't go. Maybe a smaller proportion of us would go. And so instead of doing this with people and velociraptors, I do this with worms and the thing they're afraid of, which is copper. So copper is somewhat toxic to the worms, and they learn to avoid it, or they avoid it innately, I should say. And so they avoid it on these agar plates. We put down just a drip of copper solution. And the thing that they love is the smell of buttery popcorn. And so if you ever come into my lab on a day where I'm doing one of these experiments, it will smell like a cheap movie theater. I'm sorry, or you're welcome, depending on how you feel about that. And so then we put these animals down and we let them make decisions. Do they cross the copper barrier to get to that buttery popcorn smell, or do they avoid it? And that lets us ask, basically, what is controlling risk-taking? What are some of the molecular mechanisms or states that are determining how these animals change their behaviors to do something riskier? And I really love thinking about this assay from the perspective of humanity. Sometimes we see people partaking in something we wouldn't usually do, and we have to ask ourselves, well, what's going on inside them that would lead them to make these decisions? And so one aspect that I'm going to talk to you about is this internal state that alters the sensory integration behavior. And I should say that all of this work that I'm showing you with this assay was started by Eunice Lau, a former PhD student in the lab. And the work I'm going to show you on this slide was done in collaboration with Jess Haley, who's a current PhD student in the lab. So I put some animals down, and some proportion of them get to the diacetyl, which is that buttery popcorn smell. And I plot this, and I'll be showing you a lot of graphs like this. So mathematically, all I'm showing you is the proportion of worms that get to the odor divided by the total number of worms on this plate. And we're calling it a chemotactic index. So for a well-fed wild type animal, only about 20% of them are going to partake in that risk of crossing that copper barrier to get to that volatile diacetyl buttery popcorn smell. And we can plot this. We can actually track them over time. This was done by Jess Haley, where you can see over the 45-minute assay, these animals are mostly staying right where I put them down in these brackets. Only a few are crossing. And when they do, it's usually fairly late in the game. And from this, we can get data like the velocity of the animals, where they're most likely to reside. And we have done that, and I can show you that if you're interested, but it's not included here. And then if we starve these animals for 3 hours, we actually see far more of them are willing to cross that copper barrier to get to that buttery popcorn smell. And this increases the chemotactic index by about 4-fold, where now 80% of the animals are crossing the copper barrier, or a chemotactic index of 0.8. And we can see with those tracks that far more of them are starting to venture forth toward that copper line to get to that buttery popcorn smell. And so throughout this talk, you're going to see blue bars meaning food deprived and black bars meaning well-fed. And if you've ever been hangry, I had to eat a snack before starting this talk because I get hangry. You would be very glad to know that this change, this shift in behavior is reversible. So here I'm showing you that if we starve animals for 3 hours, which is this acute food deprivation that I did on the last slide, and that I'll be showing throughout this talk, you see that after 1 hour of being put back on food, they're still behaving riskily. But after 3 hours back on food or 5 hours back on food, they're now behaving as though they are well fed. And interestingly, this is also tunable. You probably have experienced this as well, where you're not super hangry, but you're getting hangry. And we see that 15 minutes, 30 minutes, and an hour of food deprivation isn't quite enough to shift that chemotactic index. But after 2 hours or 3 hours, that's when we really see this shift. And so with this in mind, we can begin to think about what is the mechanism of this behavioral change? What is something that's reversible and tunable with timing? And this got me thinking about transcription factors. And so a few of our collaborators in the Salk Lab at SBP previously, they told us about this metabolically active transcription factor called HLH30 that is beautifully labeled using this HLH30 GFP transgene. We can see it in the nuclei of the intestine. And we can also see, and that's when it's active. We can also see when it's in the cytoplasm, a little bit of nuclear localization, or definitely in the cytoplasm, we can assume that it's inactive. And so I decided, well, maybe this transcription factor is involved in this aspect of behavioral change. So I went to see how this nuclear localization changed over time during food deprivation in wild type animals that have this transgene. And I see that well-fed animals don't have any HLH30 GFP expression in the nuclei, but at 1 hour, we begin to see some of that expression, and then it goes away. And this leads me to believe that perhaps at that 1 hour of food deprivation is when that transcription factor is entering the nucleus, becoming active and transcribing downstream genes. And to really just kind of hammer this out, I used an HLH30 mutant. So these are animals that in their whole body, they're lacking this transcription factor, and went to see if C. elegans would change the behavior when they don't even have HLH30 present. And what I hope you can see here is when we don't have HLH30 anywhere, that the food-deprived animals look exactly like the well-fed animals. This makes us believe that HLH30 as a transcription factor is necessary for this behavioral change. And its timing kind of makes sense with that tunability of this behavioral change. So next, let's think about what HLH30 is doing. So as a transcription factor, HLH30 drives expression of some insulin peptides. And in C. elegans, insulin peptides make up a large swath of neuropeptides. There are 40 insulin-like peptides, and they really allow the animals to signal throughout the body what's going on. And so we know from previous literature that HLH30 binds and is in charge of expressing some insulin genes, these 9 shown here. And so I wondered, which of these insulin peptides, if any, are involved in this behavioral change in an HLH30 dependent manner. Because remember, we have this well-fed, inactive state, 1 hour of food deprived, active state, and after 3 hours, it's still inactive. I haven't gone later to see if there's more going on, to see if maybe after 6 hours or so of food deprivation, it shifts back and maybe starts making a different signaling neuropeptide. And so again, these are making these insulin peptides. So I went through and looked at these 9 insulin-like peptides that are known to be regulated by HLH30 because they contain this E box, which HLH30 binds to. And I knocked out each one of these 9 insulin-like peptides. And what I'm showing you here is the same chemotactic index. There's just more stuff going on the x-axis. So our wild type animals always have that increase in chemotactic index upon food deprivation. And we see that same pattern in INS3, insulin 4, insulin 6. And so these are all knockouts. So insulin 10 being knocked out doesn't affect this. Insulin 11 being knocked out doesn't affect this. But we see maybe insulin 23 and maybe insulin 31 fail to behave like wild type. So this leads us to believe that perhaps insulin 23 and 31 are involved in the expression of this behavioral change. And although there's 40 insulin-like peptides encoded in the C. elegans genome, there's only one insulin receptor. And this insulin receptor is called DAF2. So what we did was we mutated DAF2 to see if we lost that behavioral change upon food deprivation. And using 2 different alleles for DAF2, we see the same thing, that these animals fail to change their behavior upon food deprivation, meaning that the insulin receptors required, 2 insulins are required, and the transcription factor required for making those insulins are required. So I think we're going into the right pathway here. And importantly, we can use transgenic tools to rescue DAF2, this insulin receptor, in specific tissue types. So here I'm showing you a rescue in neurons, which you see rescues the phenotype beautifully, whereas intestines and muscles do not. So this means that likely DAF2 on neurons is receiving that signal from the insulin-like peptides. So they're likely coming from the intestine, which is where HLH30 is active, being received by neurons that are expressing DAF2. So in summary, HLH30 is a transcription factor that binds to an E box upstream of a few insulin-like peptides, specifically insulin 23 and 31, that are received by neuronal DAF2. And this leads to a behavioral change upon food deprivation. If you're interested in more of the mechanistic detail of this, you can check out our preprint. We're currently in revision, so there's a few new experiments that aren't in this preprint, but it's at bit.ly/Matty Lab. And for my rest of my time, I guess, as a postdoc, I'm really interested in other intestinal states. So if you remember, my background is actually in host microbe interactions. And so I'm really interested in how microbes can affect animal behavior. And many of you might be scratching your head and thinking, what, microbes? Those don't affect behavior, they just like make us sick. Well, they can. And even the ones that can make us sick or make other animals sick can affect behavior. So one of my favorite examples of this is Toxoplasma gondii, which makes rodents less afraid of cats. That allows the rats to not be afraid of cat urine, and the toxo gets into the cat, which is the best living environment for Toxoplasma gondii. But even commensal non-pathogenic organisms are capable of altering animal behavior. There's some evidence that microbial colonization makes mice more social, and the microbiome of infants is associated with fear-based behaviors. And so this and many other beautiful bodies of literature support the microbiota gut-brain axis, where the microbes in the gut are allowing for signals to be sent and received by neurons. And where does C. elegans fit into this? Well, I think I mentioned that they eat bacteria. And the only bacteria that they usually eat in the lab for the last like 80 years is this boring old E. coli called OP50. It'd be like if all we did was eat oatmeal all day, every day. It's very easy to use, but it's not super ecologically relevant because in the wild, C. elegans live in rotting fruit matter. They live in these rotting apples and eat this really diverse repertoire of bacteria. So about 5 or 6 years ago, some folks across the globe started collecting wild C. elegans and getting the microbes off of them. And they found that they were filled with thousands of different species of bacteria. And they took the top 12 bacterial strains that could be cultured in the lab and made a model microbiota called CM Bio. And I've been working a lot with CM Bio. I'm not going to show you all of my data, but I'll show you one really cool piece of data on how these complex mixtures of microbes, or even just an individual microbe that's different than this E. coli species, can alter animal behavior. So you're familiar with these data where a well-fed animal, so they've been feeding on that boring old oatmeal E. coli, and they have been starved for 3 hours, so taken off that bacteria. And we see that really nice shift in their chemotactic index. If I expose them to CM Bio for just 3 hours, I see a shift in their behavior. Maybe it's a kind of a middling response, not quite well-fed, not quite food deprived. But if I treat them or expose them to these 12 species of bacteria from CM Bio individually, just for 3 hours, we see a really diverse range in behavioral responses. And I should note that each of these strains serves as food. So they're not actually behaving because they're starved. They're behaving because there's some sort of cool signaling going on from the microbes likely. So for example, this PM and MyB11 lead the animals to behave kind of like they're food deprived, as though they're risk taking. And whereas Job 134 makes them behave as though they're well fed. And so this is really cool, and others are looking into this as well. Some of our collaborators at the Baylor College of Medicine have recently shown that the composition of the microbiota is determined by insulin signaling, specifically a few insulin-like peptides and their receptor DAF2, that changes the abundance of different microbes in the gut. If this sounds familiar, it's because we've shown similar things, including one insulin-like peptide, insulin 31, that is the same across both of these, as well as their receptor. And this leads me to ask questions like, what bacterial metabolites are involved? Are the bacteria making HLH30B more nuclear localized earlier on, or is it something else? Do other behaviors use this same axis? You know, with insulin 31s both here and here, I'm wondering, what other types of behavioral shifts might be using the same pathway. And then my future lab, I hope to investigate how stable the microbiota is and how abiotic and biotic factors might shift the microbiota that might result in behavioral changes, getting at this kind of chicken and egg idea of like, what is changing behavior? Is it the microbiota, or is the behavior changing the microbiota? So I'm going to shift gears here really abruptly because I'm running out of time. And that is to share with you that I'm interested not only in the internal states and environmental effects on worms, but also on people. And so we all know that during the year 2020 through 2022, we've been dealing with some really wild living arrangements and life setups. So I started this program called Virtual Active Science Engagement to allow undergraduates an opportunity to engage with each other and with their love of science. And so Virtual Active Science Engagement, or VASE, focuses on providing students an opportunity to build skills related to reading papers, making CVs and resumes, talking about science. They get exposed to really amazing guest lecturers, you could be one, who have different life trajectories than me. And the students get a chance to network with each other and with our speakers and mentors. So if you're interested in engaging with undergraduates in a short one-off moment in your life, please think about scanning this QR code or going to bit.ly/MattyVase. And VASE has had an impact on these students. So the students, we have about 60 in the group. I'd say at any time, 20 to 30 are active, and they're globally distributed. We have students in Turkey and South Africa and Puerto Rico and all across California and India. And so before they started in VASE, I asked them, how prepared do you feel to have a career in STEM? And most of them said, between a 2 and a 3 for preparedness. And after just 10 weeks of VASE, after just our first session, and now we're in the 6th term of VASE, we shifted their feelings of belonging and preparedness to between a 4 and a 5, which is pretty amazing. We've also changed their behaviors. So students are spending more time thinking about their future careers, reading scientific literature, and networking with peers, just after a 10-week program. And this is all optional. Students come and go as they like, and it's free and it's open to any student. So if you have an undergraduate or even a senior high schooler in your life who you think could benefit from this type of engagement, send them my way. And if you're thinking, man, I really want to get involved, I really want to do some things, you can help by giving us some of your data. So these students really, really want to do research, and doing hands-on research is one of the best ways to increase sense of belonging and persistence in STEM, especially among women of color, which are the primary students that are involved in VACE. So you can sign up to be a virtual IDI project mentor, where you'll introduce a topic of research, share a little bit of data, and show a student or a group of students how to analyze those data. The students will work independently, send you their results, and take a little bit of their own time. They get hands-on experience working with data, they learn a new field, they network with you, they feel confident. It's really outstanding, the outcomes of these projects. And you get experience of mentoring without a huge time investment, and you might even get some of your data analyzed for you, and blindly. So if you have like 1,000 images where you need some red dots to be circled and counted, like this is perfect for you. Some of the students who have participated in virtual IDI projects say they learned patience and time management, they learned how to use ImageJ to analyze mitochondrial DNA, and that they learned that publishing research means you have to analyze a lot of data and have time management skills. So these are really amazing skills that I didn't learn until middle of grad school that you could help a student learn now. So again, if you're interested in a virtual IDI project, you can scan this, and I'll post some links in the chat once I'm done yammering on. So finally, thank you all for your time. I'm at the Salk Institute in the Chalasani Lab. We're a great group of people. And like I said, this project that I mostly talked about was started by Eunice Lau and supported by Naya Brooks and Karina Kono. And I work with an amazing undergraduate in the lab, Nadia Hagani, who gives my life meaning most days. I have amazing collaborators across the globe, and I received funding from the postdoctoral research fellowship in biology. If you're interested in applying to this, let me know. I'm happy to share my materials with you. If you are a female or non-binary postdoc and you're interested in a faculty career, consider applying to the Leading Edge program. I'll pop a link to it in the chat. The application is due in February. It's an amazing group of young scientists. And I have some other affiliations and mentoring opportunities. If you're like itching to get involved with mentorship, highly suggest looking at NSERP, Scientifico Latino, Bump at UCSD. And thank you all to Dazzle and all of you for listening.